This is an A-level biology presentation looking at digestion and absorption. So to get started, all you have to do is cast your mind back and identify all the parts of the digestive system. Pause the presentation now to give yourself some time to come up with your answers. When you're ready, you can restart and check how you've done. So pause now. Well, these are the names of all the different parts of the digestive system and all the different sections of the small and large intestine. Make sure before you go any further that you have memorized all of those parts. So in this session, we're going to look at the digestive system. We're going to look at digestion by certain enzymes. We're going to look at bile salts, and then for cotransport and the role of micelles in absorbing different substances. So digestion can be split up into two parts. We have the chemical digestion that involves enzymes and substances like hydrochloric acid. And we have the physical digestion, which involves the use of a range of muscles around the body, as well as obviously things like teeth. Remember, you should have already covered properties of enzymes and characteristics of enzymes in terms of being proteins and polypeptides, and make sure you go back over the section of biological molecules should you need to. Carbohydrate is broken down by a group of enzymes called carbohydrases, and they break those carbohydrates down into what we call simple sugars, which for A level we refer to the phrase as monosaccharide. Quite often the more complex sugars are actually broken down by a series of enzymes. The classic one, of course, from GCSE is that starch is broken down into maltose using an enzyme called amylase, and then maltose is broken down into a glucose molecule by an enzyme called maltase. And that digestion occurs in different parts of the body, because remember, amylase can be produced in the mouth, salivary amylase, and is also produced by the pancreas and then released into the duodenum of the small intestine. In terms of protein digestion, we use a group of enzymes called proteases. And remember, proteases convert proteins into amino acids. Just be aware there has been a couple of examples of exam questions that have focused on proteases as groups of enzymes, and you had to make comparisons between the endopeptidases, the exopeptidases, and the dipeptidases. Remember, if you put endo onto the start of any enzyme, what it does is it breaks peptide bonds between the amino acids inside the long chain of the polypeptides. Exopeptidases only break off the very last amino acid from a chain, normally at both ends. And of course we have our dipeptidases, which hydrolyze the bond between two amino acids, but they are membrane bound to epithelial cells in the ileum. And that's an important point to remember. It is on the specification. It's a little point, but it could get you some marks. So the dipeptidases are the ones that are membrane bound on the epithelial cells lining the ileum, and we will talk about the ileum later on. The final group of enzymes I'm going to briefly mention are the lipase enzymes, which of course hydrolyze lipid. Lipids are broken down into glycerol and fatty acid. The lipase enzymes are of course produced in the pancreas, and they hydrolyze the ester bonds between the glycerol and the fatty acid. They will form into droplets called micelles, and for that to occur we need to have a process known as emulsification using bile salts. So the primary purpose of a bile salt is to increase the surface area of a lipid and allow the speeding up of enzyme reactions with lipases so that they can then absorb the fatty acid into the body. We'll go into that more detail later on about the actual processes and mechanisms involved in doing that, but just be aware you should be clear on the concept of what emulsification is and what an emulsifier is. So that's a brief run through the digestive part of the system. What we're going to now look at is the absorption part of the digestive system. To start with, we have the ileum, which is the small intestine, and that's the bit where the majority of the absorbing into the bloodstream occurs. There's a number of characteristics about the ileum you need to be aware of, and not only do you need to be aware of those structures, but you need to be aware of how they relate to Fick's law. So pause the presentation now, Look through each of the structures that you're given and try and identify how they apply to Fick's law. When you're ready, you can restart the presentation. So pause now. Well, remember Fick's law, we talk about surface area, we talk about concentration difference or concentration gradient, and we talk about the diffusion pathway. And all of these things can be used to affect the rate of diffusion. In the second half, what you were asked to do was link each of the characteristics, the structures, and explain how they're adapted to their particular functions. When we transport substances from the ileum into the bloodstream, one of the most important ones is, of course, glucose. Glucose is the main source of energy during cellular respiration, and as a consequence, it's extremely important to the body. If we had diffusion of glucose into the bloodstream, the problem we've got is that at some point where we've done very little exercise and we have eaten very little for a long period of time, the levels of glucose inside the blood capillaries could become higher than those found inside the small intestine or the ileum, and as a consequence, glucose could move back out of our blood capillaries by diffusion, which is obviously not something that the body wants for something as important as glucose. So the mechanism for absorbing glucose is known as cotransport. Now, cotransport is a specialized type of diffusion, but it's actually dependent on active transport. And what you've got is you've got a thing called a potassium-sodium pump. The potassium-sodium pump actively transports sodium out of the cells that form the lining of the small intestine. As a consequence, we have a small concentration of protein. And as they move through the cell membrane using a cotransport protein, we have different types of proteins. Earlier we mentioned the fact that lipids are also absorbed into the body, and we also mentioned about bile salts. Well, there's a specific process you need to know about how we get lipids absorbed into the bloodstream, and it's a little bit more complicated because unlike glucose, we do not absorb directly into blood capillaries. We actually go into the lymphatic system. For start off, you have to remember that we have these things called triglycerides, which is another term for a lipid. A triglyceride is made up of a glycerol and 3 fatty acids. But we can break that up into what's called a monoglycerate, which is where we have a glycerol molecule and we have only 1 fatty acid attached, and that leaves 2 fatty acids on their own, and that can be converted back into a triglyceride under the right circumstances. What we're going to do now is we're going to look at how we get a triglyceride from the lumen all the way through, eventually into a lymphatic vessel, and then eventually into the bloodstream. What I'd suggest you do at this point is pause the presentation, and if you have a phone or other device, take a picture of what's on the screen with all the labels, and you'll make it much easier for yourself to follow as we work our way through the rest of the presentation. So we'll start with, we've got our lipase enzymes, and the reason we're using lipase enzymes is because we're breaking down a lipid. We use the term hydrolyze instead of the word breakdown. And remember that we are going to form a monoglyceride and 2 fatty acids. Well, the monoglyceride and the 2 fatty acids are then able to associate with a bile salt, forming a micelle. And the micelle then makes contact with the epithelial cells that make up the wall of the ileum. They're broken down and they release the monoglyceride and fatty acid. And as the monoglyceride and fatty acid are nonpolar, they're able to diffuse across the cell surface membrane, and they move into epithelial cells that form the layer of the lumen. What then happens is we then have endoplasmic reticulum, And we then form what are called triglycerides, where the two fatty acids are stuck back together with the monoglyceride. We then progress on, where lipids and proteins are then added, and then what we have is the Golgi apparatus associates with cholesterols, lipoproteins, and everything else, including the triglyceride, to form what's called a chylomicron. And a chylomicron is a specific little structure that's adapted for the transport of lipids. The chylomicron is then able to leave the epithelial cell by exocytosis, and it goes to what's called a lacteal, which is a structure found on a lymphatic vessel. From the lacteal, the chylomicron then moves through the lymphatic system, and the chylomicrons are hydrolyzed by enzymes inside blood capillaries. So the lymphatic vessel, remember, feeds back into the vena cava next to the heart, and then obviously it'll circulate around the blood system from there. It gets broken down back into triglyceride, and the triglycerides are then able to diffuse into cells. So this is the entire process, step by step. You may want to use the images, you may want to add your own images, tweak it whatever way you want, but you need to be able to go through that process step by step. You need to know what micelles are, you need to know what chylomicrons are, and the sequences of stages involved in getting triglycerides from the ileum all the way into the blood capillaries. Just to finish off, make a list of all the possible exam questions that could be asked that are linked to this lesson. Remember, you should use the exam STEM words describe, explain, and compare, and then spend a few minutes and try and identify how would you answer each of these different types of questions differently. So how would your answer to a describe type question be different to your explain or be different to your compare?